This biodiversity of wild food and medicinal plant of Eastern Himalayas was a topic panelized not by me but my student who were working as a PhD scholar last time. And here I just would like to briefly just highlight a few findings of the research of what we have been conducting since last 5 years as ethnobotanist. And at the same time, at a field level, at the laboratory level, we have tried to validate those in the phytochemistry, the value addition of those food and medicinal plants which are available mostly in the Eastern Himalayan region. We know that those parts are very rich in biodiversity. And we had another interest on that. The food and medicinal plant have a lot of, the compound of very nutraceutical interest, that, but then just for the production and just to garner some industrial scale production kind of things, we need some kind of expert like a plant physiologist who really could understand, agrotechnology and then value addition cultivation, so that those kind of secondary metabolite which we are looking for could be maybe improved because of this true stress induction or some kind of things. So with the help of this Mississippi State University, we started working on that. The Professor Reddy was enough to cooperate something on that line, and just since last 5 or 6 months we are working. But this is very a preliminary kind of talk that we are going to deliver. So this region is very rich in cultural and biological diversity, and very unique in the sense the top 12 mega diversity hotspot. Then we have the highlight here. The two aspects, the ethnobotanical aspect of some traditionally used food and medicinal plant. Number two, apart from traditional food and medicinal plant, just we have some biochemical studies on those. Then particularly the potentilla, the Vaccinum glaucum, album, Drymeria, Propinqua, Roja, Serica, Quercus gripiti, and Quercus semicarpifolia. Those are widely used by those local communities as basically as a food, but those are somehow act as even a medicine maybe. So just out of curiosity, we just went on to find out. Our basic target was to look for some antioxidant compound. Plants have thousands and thousands of compounds that we know, but here our target is only very few compounds, maybe 2 or 3 compounds, especially the gallic acid, the quercetin, and we have another compound called asiaticosides, which is found in different plant, but the most predominant in Central Asiatica. So let's see so on ethnobotanical basis we have this is the study area we have conducted. The wettest spot, the highest rainfall region, which we call the Eastern Himalayas, the state of Arunachal Pradesh, the Nepal and Bhutan, and the part of Assam. Those are the region that comprises the Eastern Himalaya actually. And we have the work with the local communities, particularly the Nisi of Arunachal Pradesh. That's the Donipolism is their cultural practices that we call, but rich in traditional, knowledge related to utilization of, especially the plant bioresources. And with this tribe that we could able to identify some kind of plant like total 187 species, mostly edible plant. That's the wild plant. So then now we have a number of medicinal plant out of literature review. Then we came to know that most of them are even a medicinal plant. It's very interesting. Then. So now the roots are even the most widely used plant and whole plant, something like this. Then let's go ahead with. So a kind of biomedical, ethnobiomedical studies we call it, just a random pill survey. So here we could see that a lot of the species. Especially, we have like the stomach-related disorders. The plant utility is very high, this kind of. So those are the richness how the plant are widely used by the local communities as a medicine, and this is now out of those, several plant. We have prioritized very few species. We have came down, screened down, just to look for very few compound like our target, the antioxidant compound. So Potentilla, Vaccinum, Drymeria, those are very edible plant. It's all are edible. So this Centella asiatica is commonly used as a salad or vegetable. Now let's see how. So antioxidant compound, it's widely debated since yesterday, yes. So it's a compound that built to defeat free radicals, you know. So our target antioxidant compound from traditional wild food and medicinal plant, the gallic acid, the basically phenolic origin, the known for anti-cancer, the anti-diabetic and anti-inflammatory activities. Then we have a quercetin, the flavonoid group of a compound, which are widely found in all the plant. Probably in the onion group, it is the highest. Say quercetin, even this gallic acid. Those are widely found in most of the edible plant. But what about the wild plant, which we never know, and most of the tribal communities they use? So we can have that source of things in addition, like. So this is how the inclusion. Now, this is just the chemistry of gallic acid and quercetin. And so those are the plant which we have selected. The first is, I think, Drymeria. Then second one is Propinqua. Yes, Propinqua. This is Propinqua, yeah. Potentilla. Then Rosa sirica. The Vaccinum glucoalbum. This is the wild, very, very sweet in nature. And the Quercus semicarpifolia and Quercus, griffithia and semicarpifolia. So this Quercus species, the fruits, they are mostly used by those Eastern Himalayan tribe as a payment food. It's mostly during when food stock is exhausted. So then that time this corn is actually used as the food supplement. So this is very interesting. Then this is used as a berry and just a commonly eaten wild berry. So then this just led us to some sort of curiosity that the compound may be some interesting compound we made. Then we bought some kind of we started estimating. So phenolic compound estimation. Then Potentilla is showing some kind of highest than Quercus semicarpifolia. Then just the flavonoid content. So even this Vaccinum glucoalbum is showing very, the remarkable the result. Then just the DPPS assay of the antioxidant free radicals. So it's especially this Vaccinum glucoalbum is showing a very good, the progressive result. So this gives us an indication that those plants can have the anti-inflammatory and anti-cancer activities. Yeah, just that may be consumed as a. So then regression curves. So ABTS. So here we have again the Vaccinum is something very remarkable plant. So and apart from that we have a Quercus semicarpifolia. Then so after that what we have done is HPTLC quantitation of quercetin from Quercus by using the markers, and then those are just the standardization which we require is just to detect that. So these are the quercetin. This is the marker standard, and from I think Vaccinum glucoalbum we have this kind of So we have the range of quercetin. Yeah, this is through HPTLC. Then this is another gallic acid from Rosa sirica. Then the Quercus griffithia is all from gallic acid, the source of gallic acid. Then this is semicarpifolia, that's the peak of gallic acid is quite clear in HPTLC, So now we can have the comparison views like chromatographic standard and the fruit extract of Rosa sirica. So here, we have a standard marker, and this is a sample track. So we have a gallic acid here, which is equivalent to your standard, or just it's indicate that something is there. So this is how the significance of this study is. And then we have there's a Quercus griffithia even. So again, this gallic acid is very much prominent. So in comparison to standard, so this is also indicating how remarkable it. So then this is very interesting, another gallic acid peak, which is very much comparable to the standard. Yeah. So that means those plants, especially the semicarpifolia, and other groups have a lot to say about antioxidant. Yeah, so this is a kind of standardization, especially when we talk of herbal drug. So we have to somehow standardize that this compound, the RF value, this much is this, so that in future, if there is any kind of adulteration, so we could easily detect it. So this could play a big role. So now the proximate composition is also not bad, especially the carbohydrate is very high in all plant. That means it's a consumable. So this is basically a kind of edible plant, and this is just the crude fiber is also good enough. Then yes, mineral element. So potassium, the nitrogen, the calcium is very much it's maybe within tolerable limit of a biological system. And then the vitamin always is required in a very traces by the human biological system. We don't need actually lot of vitamin in our body. What every vitamins, maybe A or B or C, actually we need in a very traces amount. So that's how it's. So we have a lot of, say, carotenoid. So mostly carotenoid probably mostly is the vitamin A. Then alpha tocopherol is mostly vitamin E group, which help in cell membrane stabilization, and it's a powerful antioxidant. So consider as, and at the same time, this ascorbic acid is also a very important vitamin. So those are just found in traces, but could cause a significant physiological, changes in human physiology, and this is very important because vitamin often act as a hormone, we call it. So that means you don't need a very big amount when it is called hormone. So we need a very small amount so that it causes marked physiological changes. Now, this is anti-nutritional factors. So it is very interesting to know that all the wild plants do have that kind of anti-nutritional factors. It is only because it is nature's built actually to be a defense mechanism for those plants actually. The defense mechanism is essential for those, maybe the wild plant, from grazing by the animal or insect, that is very important. So that kind of tannic acid, phytate, oxalate, saponin, those are actually a kind of for us, from nutritional point of view, it could be anti-nutritional, but traces amount is required in every plant. So it's, but we can say those are within tolerable limit of biology. But after necessary processing, those can be consumable. So there has to be some technique, developed technique to remove those maybe tannic or phytate or oxalate so that it become more consumable. That kind of things can be done. And this is just one of my students. He have conducted some pharmacological kind of activities. And taking the paracetamol, this is some kind of analgesic, I think, drug, just to see how this elevated body temperature can be reduced by the plant extract, particularly Bixium glaucoalbum. So the result is very significant in the sense that, so this is the standard, this is standard, this middle one is standard, the paracetamol drug, and those at least the plant extract, the blue and red, those are actually proximity. That means the walking, somehow walking. So this shows that the plant could have anti-inflammatory or antipyretic activities. Antipyretic means something, that reduction of the body, elevated body temperature. So now, in our key findings, what we have seen is that there is a presence of antioxidant compounds such as quercetin, which are mostly of flavonoid origin, gallic acid, then carotenoid, ascorbic, alpha tocopherol, vitamin E, which are most widely predominantly found in those wild plant. And even those are mostly common in most of the cultivated wild fruits and vegetables that we consume daily. So proximate analysis also shows some kind of nutritional potential that plant do have, but we have some anti-nutritional factors like tannin, phytate, and oxalate that could be made consumable after necessary the processing, and that could be within our biological toleration limit of biological system. And here is what interesting. I'm just sharing very brief. The fruits of Bixium glaucoalbum contain a flavonoid compound that is called quercetin, as per our estimation, which is much higher than teas, red onion, and apples. So we have a 280 mg per kilogram of dry sample, just in case of vaccinum. So in tea leaves, it's already known. Internationally, this standard is known. And red onion, we have a 13.27, and apples, we have 4.42, the mg, of per kilogram of, in apples. So this is how this plant could be very useful in sense. So then another compound is the gallic acid, which are widely, predominant, the compound that we have witnessed in Quercus and the Rosa serica. And in Quercus, we have just seen very, lot good quantity. So then finally, we come down to our one of very interesting, but this is ongoing, very preliminary, very ongoing. And this is just a very visible result that our ongoing experience shows. So we are looking for asiaticoside, which is, the brain memory booster, people called it in traditional Indian medical science. So a specialty of this asiaticoside is it's a pentacyclic triterpenoid saponin, the antioxidant agent for brain and neurosystem. So those are already reported in traditional medicine like Ayurveda. So cure dementia, Parkinson, and there are lots of literature there since compound has been isolated and work has been extensively done. But in terms of stress physiology and this, no work has been done yet. And in terms of agro-technological development, but plants are widely consumed by local communities, especially the Asiatic communities. So plants are quite common in South China, Africa, India, it's Sri Lanka, very much common plant and widely used plant. But a specialty is that it is traditionally used as a brain tonic. So who do not like to become maybe the Einstein? So maybe everybody curiosity is there. So that's why there has been a lot of interest over this plant. So at Mississippi, we have tried this kind of experiment, which is quite visible. This is just a 1 month, just observation we have kept on the greenhouse. So this is a control 100% water and 100% nitrogen. Plant is very healthy in 1 month, and in drought stress situation, 50% water, 100% nitrogen, little study, nitrogen stress, 100% water, but it seems little healthy still, but drought and nitrogen stress, just a little stress. So our just target is to have a lot of studies out of this experiment, especially from the leaf. We are going to analyze the phytoconstituent, the increment or the decrease maybe. So especially in the stressful situation, it is well assumed the fact that secondary metabolite production become much higher in stressed situation. It is all assumed. So now we are going to, in lab, we are going to see that how, at what percentage the asiaticoside level has increased or decreased, or what new kind of compound could be, seen out of. So just this morphological measurement, the root, growth measurement is just a normal experiment that we are going to do that. But at the same time, our primary target is to look at the asiaticoside, the concentration, the increase or decreased at a particular hour of treatment or a month in that way, so that we could develop a standard in that sense that asiaticoside from this plant could be, improved through better cultivation practices. It is WHO mandatory, there is a guideline that good cultivation and good harvesting practices there is. So for every species of a drug kind of a plant, we need to develop that kind of standard experiment. So with that objective, we are just moving, and it's a very preliminary experiment. I'm very sorry. So this is just another that was without UV. Now we are just giving some UVB stress here again in this group of plant. So again, we'll just try to look at the secondary metabolite, that whether the secondary metabolite is going to. So we are talking of the global warming and climate change, and plus that depletion of the ozone layers that would actually lead to lot penetration of UVB, this kind of radiation. What about the vegetation, the secondary metabolite production? Will there be any usefulness of that kind of radiation? We are just out of curiosity looking at that angle, and this is very interesting. So this is just the preliminary kind of things, and we are just looking at that. So this is a very healthy plant, just 100% nitrogen and 100% water, just a very healthy, happily growing plant, the Asiatic. So now with this, I think just a very brief sharing. Nothing I have to. So thank you so much for your patience sharing, and I'm very much thankful to those my PhD scholar and the colleague Dr. Pallavi Kalita from National Institute of Technology India for doing the biochemistry, and Dr. B.J. Gogoi, DRL, Ministry of Defense, Government of India for his phytochemistry and other analytical work, and Dr. Raja for supporting me in this physiological work and understanding how to build phytochemistry and physiology together so that there is a holistic understanding of the situation. So genetic level studies, I could not find any partner yet, so maybe in future I just try to look about how this gene regulation kind of things can be done in future. Those are just the scope, that just the sharing kind of thing. So finally, I just my funding agency, the Ministry of Defense, then the University Grant Commission, Government of India, for their valuable support throughout this 5 years of studies. So thank you very much. Any questions?